So thank you everybody for coming to another seminar. Today we have Òscar Guadayol. He's a postdoc researcher at So thank you everybody for coming. Así suena mi voz. No sé por qué nos escuchamos cuando somos nosotros que transmitimos la audio desactivada. Sí, pero si no comenzaría a hacer. Sí. Bueno, ya fue. Today we have Òscar Guadayol. He's a postdoc researcher in the UIB group, and he will be talking about not fish, people like fish, so about Phytoplankton. It will be interesting. So thank you very much, Òscar. The floor is yours.
Yeah, I'm sorry I'm talking about fish, and I'm going to talk about arguably most ornamental fish. Yeah, and I would like to start. I usually like to start with this plot here. This is a classic plot. It's a size spectrum of biomass in the ocean. And just to reiterate how important plankton is, it shows the abundance versus the body mass in logarithmic axis, no? And what this highlights is the fact that biomass is very much biased towards the smallest sizes, no? The smallest organisms are the most abundant. And not only that, but also there's a bunch of different relationships that show how it's also the size class that carries most of the metabolism of the ocean. Small organisms are not just more abundant, but they are more diverse. They are faster at doing anything, at processing energy and incorporating matter, etc., etc. So that's one of the reasons why I study this. And one thing that is, I think it is a little bit surprising, is the fact that the physics that these very small organisms experience in their daily life are somewhat non-intuitive to us, because we live in a world that is where inertia dominates, no? But it is not the case for microorganisms. Here I have a very striking example of this. No? It has been calculated that if a small bacterium was swimming and all of a sudden decided to stop and the gel stop rotating, it would coast for about 0.1 angstroms. That is about 1/5 of the diameter of a hydrogen atom. So it is that quick. There is no inertia at all. And that creates a completely different physical environment to which we are used to. I think this is best shown with this classical Reynolds number, which is basically a ratio of the inertial forces versus the viscous forces. We live in this world where inertia is very important, where we have turbulence and transport of matter is carried out mostly by turbulence and mixing of waters, etc. Microorganisms live here, where a flow is really not very important at delivering molecules, nutrients to their surfaces. No? And so viscosity is very important to these organisms. And just to clarify and we all have a little bit of an intuitive idea of what viscosity is, no? But just to nail it down, viscosity it can be defined as the measurement or measure of the resistance of any fluid to being deformed, to being moved. And it has a bunch of different consequences for the life of microorganisms and their life. Like starting with what I was mentioning, no? That the molecule, the transport of molecule of matter is not done. It's not done by inertia. It's done by viscosity, and so it's diffusion. It means that it's very, very, very fast at really small scales, but it's extremely slow at much longer scales. The other interesting fact about the physics of the microbes is that any particle at these scales is subject to Brownian motion, which is basically this random motion that particles and molecules experience because of the collisions, the constant collisions with water molecules that they experience. And then the last thing is that, and we've seen that with the example of the coasting bacterium, is that they seem to be swimming, microbes, as if they were in a sea of molasses. If you want to imagine how swimming as a microbe is, you have to imagine yourself swimming in a pool of honey or molasses. So viscosity is very important for microbes. It affects all aspects of their life. And the question is, okay, what do we know about it in the ocean? How is viscosity changing in the ocean? And the answer is that we know surprisingly little, given the importance of microorganisms and the importance of viscosity of microorganisms. And this is because from the point of view of physical oceanographers, it's a very interesting question. They figured out many decades ago that you can calculate the viscosity of seawater if you know temperature, the salinity, the depth, and that's it. And it's perfect equation, and you can actually, that would be it. We just took a climatology from a random year and from seasonal phase, temperature and salinity, and calculated viscosity. And as you can see, the variability is quite low. So it's not very interesting to them, but to us biologists, it's very interesting because the fact is that seawater is not just seawater. It has a lot of different compounds, and all these compounds have biological origins. And this is something that we are realizing more and more nowadays, no? That there is a huge variability in sizes, but also in diversity and complexity of organic particles and macrogels, transparent exopolymeric particles, then all the way down to microgels, colloids, macromolecules, all the way down to dissolved inorganic molecules. And to me, the interesting part here is that all these molecules in the water are going to have an effect on the physical properties of the water itself. And this is something that we are aware of. A few decades ago, especially with the executives and collaborators, they realized that there are some phytoplankton species that when blooming or when grown in cultures, they release a lot of stuff that increases dramatically the viscosity of seawater. And they've seen this at the mesoscale. So, for example, I have here plot of chlorophyll and seawater on a time series of few days, a couple of weeks, I think, and you can see how viscosity increases, matching a little bit the increase in chlorophyll. And we've seen also these at fine scales. This is centimeters. From 0 to 45 centimeters, we have the same chlorophyll and see whether there is viscosity that matches what we see with chlorophyll. The question to me is, well, given that where the effect of viscosity is going to be felt more strongly is at the microscale, what happens at the microscale? And here I want to just to introduce also this kind of new paradigm that we are experiencing, this paradigm shift that we are experiencing in the last couple of decades, from Kazan's hotspots, where we envisioned that the ocean at the microscale was actually quite heterogeneous because of the high diversity of different organisms that were interacting with each other. And then Stocker's adding this concept of zero gradients, where he hypothesized that all the activity of these microorganisms were also generating huge heterogeneity in chemicals. So we want to add also what would happen also in the viscosity of this picture, because in the same way that the chemical variability, the activity of organisms is affecting chemical variability, we hypothesize that it's also generating gradients of physical properties, and in particular viscosity. And it's not a new idea. It's an idea that has been mentioned from time to time in the literature as something that could be possibly happening since the 90s. But it was never actually tested because we didn't have the tools until recently. So we realized that there was this available tool. It's called multiple particle tracking microbiology, which is a technique that is very, very much used in biomedicine to measure the rheological properties of cytoplasm and of plasmodes and things like that. But had never been used in oceanography, and it's a very basic, simple technique, and it takes advantage of the fact that particles move very predictably in a Brownian motion that we can characterize. And this is described as this Stochastic equation that relates the diffusivity of these particles with temperature, size of the particle, and viscosity. Okay? So by seeding your fluid of interest with a bunch of spheres of known density and known size, and at the control temperature, if we can characterize their diffusivity, we can characterize the viscosity of the fluid in which they are immersed. And doing this, we can actually create this kind of match of viscosity at very high resolution. Saying that these bits here are 300 nanometers in diameter, and they are fluorescent, so they are very easy to track. So we do this. We just have our bunch of different Phytoplankton cultures, and then we see them with all these fluorescent little beads, and we track them with a microscope. And from here, we can generate this kind of maps of viscosity, which show us several things. The first thing that was apparent from the beginning is that viscosity is quite heterogeneous, much more than we anticipated in general. The second thing is that in one of the most important hotspots of microbial activity, which is marine particles, marine snow, viscosity was extremely high, an order or more of magnitude than seawater, pure seawater. The third thing, which is what I'm going to focus, that we realized is that around phytoplankton cells, and precisely because they were exuding a lot of exopolymeric substances, EDS, you could find gradients of viscosity. So this is a diagram cell, Phytoplankton aphides, and this is a profile of the viscosity away from the cell surface. You can see how as we approach the cell, viscosity increases sometimes quite dramatically. And it's something that we observe over and over and over again. This is very interesting because this area here that we call this area that surrounds the phytoplankton cell, which we call the phycosphere, is also a hotspot of activity. That is where many of the interactions with other general microorganisms and in particular with bacteria are occurring. And these are interactions that are carrying out a huge proportion of the metabolism of the microbial community. And therefore have importance. Understanding this is important to understand the nutrient cycles in the carbon cycle and the effects that these microorganisms will have on the way up to the climate. So this example here, let me show you, is it working? This is a cell of diamonds, and it doesn't seem to be working. But you can probably see all these small dots around it. These are bacteria. And this is something that you see when you look at the phytoplankton cell in the microscope, you are seeing surrounded by bacteria swimming around. Okay, so as I said, all of this kind of increasing viscosity is caused by the exudation of these exopolymeric substances, which are a complex mix of proteins, polysaccharides, and other stuff. But one thing that is interesting about this is that the effect that it has on the viscosity depends on how you are measuring it. So if you are using large beads, these beads will get entangled in this matrix of polymers and will not be able to move. Then when you look at the track of the bead, it will look like it's normal bead, so the viscosity that the particle is experiencing is very high. On the other hand, if you are tracking a very, very small particle that can fit in between the pores and the network of tubes that we have here, you will see it moving quite fast, almost as if there was nothing. So the viscosity that you would be measuring would be quite low. Meaning that the apparent viscosity or the effective viscosity depends on the size of your particle of interest. And this applies not just to my beads, but it applies also to bacteria, it applies to macromolecules, it applies to nutrients, organic nutrients, right? And so this made us think, okay, what if the explanation of this stuff is actually smart strategy that allows phytoplankton to kind of gain a control on their resources. So they can trap bacteria that are good for them because they can interact with them and they can establish a serious relationship with them. They can also reduce the diffusion away of molecules that can be interesting, like infochemicals or exozymes or organic nutrients. And at the same time, it may not have a very large effect on the uptake of inorganic nutrients because the diffusivity of these organic nutrients is not really that much increased. Right? So that's our kind of underlying hypothesis for what I'm going to show. And this is all work in progress. So so the first thing that we were looking is how are these layers of viscous stuff going to affect the diffusion of organic materials. Now, we did this with a very simple way, with a tiny chamber, to look at how the concentration of, in this case, present day decrease in the case where we have just seawater or seawater with viscous stuff. And you can see how gradients become much steeper and they dissipate much slower. We also corroborated it with numerical simulations. In this case, I'm showing this is a simulation of a phytoplankton cell surrounded by seawater, and this phytoplankton cell kind of breaks. So it releases all the phytoplankton, all the cytoplasm. Okay. And so here we have partition gap on the top. What will happen if the cytoplasm at the same viscosity as seawater? And basically you see after 5 minutes it's diffusing away very nicely. Okay, but if the cytoplasm was viscous, which is more than likely, what would happen is that it would take much longer for the stuff to diffuse away. I mean, it would be retained in the vicinity of what was the phytoplankton cell. This has implications for the interaction with bacteria. But this does not answer the question: is what is the is it really going to affect that much the diffusion of small molecules? This layer of viscosity, and this is something we are working on now in several ways. We are using microrheological to actually characterize the viscosity and at the same time measure different diffusion of different molecules. We are also looking at combining analytical models and numerical models to see how this is going to affect the diffusion and the shape of the chemical gradients. That's still work in progress. The other big question is how is this viscosity layer going to affect the motility of microorganisms that live around the phytoplankton cell? As you may know, bacteria move in a very particular way, precisely because of the effects of the role of viscosity. They usually, typically, they run more or less straight for a few micrometers, and then they will change direction, stumble or they can reverse it. It depends on the species, no? But basically, they do this: run, change direction, run, change direction, run, change direction, run. So we looked at how the increase in viscosity affected this pattern of movement, how it affected the swinging speed, change in direction, and how it affected the ability of running straight, for example. And you can see it here. This is a bacteria swimming in low viscosity, and this bacteria swimming in high viscosity. In kinds of different experiments, we actually subject these bacteria to a gradient of viscosities, and we characterize them pattern using video microscopy. Okay, and so we established the functional relationships of bacterial motility with viscosity. In swimming speed, which decreases quite dramatically with viscosity, swimming directionality, the ability to run straight, which is improved actually when viscosity increases, and also the thermodynamics, which it's a little bit hard to read these plots, but basically what this shows is that as you increase viscosity, bacteria cannot change direction as easily as before, so they run straight. Okay, so that all these relationships will be inputted in an individual-based model, which basically simulates the bacteria swimming around, basically, no? And also it simulates the chemotactic behavior, their ability to detect nutrient gradients, track them, and go to the nutrient source. Here's an example: is the color, the yellow, black, and color show the increase in viscosity, and then you can see the bacteria that are in low viscosity are swimming quite fast, but as they approach the viscous layer, they start swimming slower. So the end effect of this is that they are going to concentrate, accumulate in the areas where it's high viscosity. So we did these simulations around the Phytoplankton cell. And we did it in different conditions, So conditions in which gradients decrease business as usual, conditions where there were no nutrients, but there was a gradient of viscosity, and then a condition in which there was a gradient of nutrients and a gradient of viscosity, which were interacting. Okay? And this is the result. This is the bacterial abundance versus the distance from the cell. When nutrients there was a nutrient gradient that you would expect because they were exuding nutrients at a constant rate, you would see how bacteria would accumulate after 10 minutes quite nicely near the cell. About here that I can discuss later if you want. When there were no nutrients, none at all, bacteria would still increase. That's what the technique called viscous strategy. It's caused by the gradient of velocities involved by the gradient of viscosity. And finally, when you have both the gradient of viscosity and the gradient of nutrients, this increased quite dramatically. So the fact that these cells are exuding viscous stuff helps recruiting bacteria to their surroundings. And that's it, basically. I'm still working on all this stuff. Several things that then I haven't enough time to talk about, but we are trying now. For example, we are working on characterizing the physical properties of these EPS with a little bit more of detail, looking at things like viscoelasticity and absorption. We are also interested to look at how is this looking in the field. Is this real? Because it is an important step to establish the ecological relevance of what we are finding. Then also we are working with microfluidics to have an experimental confirmation of the results of the IBM that I just showed you. And finally, we're also exploring new ways of measuring the size dependence of efficiencies that is happening in the world. But just as take messages, I just stress that microorganisms generate heterogeneity, physical, not just chemical, but physical heterogeneity at microscale. That it's nutrient transports, there is no Phytoplankton cells, aggregates, etc., coexist with viscosity gradients. And at the most basic level, these viscosity gradients are going to affect the diffusion of nutrients, the creation of gradients at their shape, and microbial motility. And finally, this is what we are working on. The hypothesis that EPS is actually the tool that Phytoplankton can use to resource partitioning and to structure their environment. Just as the last idea, I focused on the effects of this viscosity on basically the microrheological. But I'm quite aware that the effects of viscosity can actually transcend these small microrheological and have effects on many different processes in the ocean, like viral infection or sedimentation of particles, or the scalar turbulence, the interaction with between corals or fishes and pathogens, things like that. So if you are interested in any of these topics or in any of these is relevant, I'm more actually to enter a philosophical discussion with it. And that's it. Thank you for your attention, and thank you all my numerous collaborators for everything you received. Thank you.
You talk about bacteria in general, but I guess you have different types of species of bacteria.
Yeah.
Do you know if they behave differently regarding the viscosity?
Yes. Yeah. It's in fact, I mean, I simplified it a little bit because this topic is quite complex. There are bacteria that will actually increase their efficiency, their swimming efficiency as you increase the viscosity. That depends on the shape. Mostly, so bacteria that are phytoplankton will swim faster, which is why many pathogens that enter our bodies through the stomach, for example, to have these helical shapes. It will also depend on the size, number, and position of the gel. So it's quite difficult to answer this. But most of my expectation is that most of the relatively simple shaped isolated bacteria that we would find in the ocean are going to have this kind of relationship.
I was more wondering whether different bacteria will go to different viscosities surrounding these cells. So whether you might have different communities of bacteria surrounding different types of phytoplankton. Not sure if at the individual level, because I'm not sure how much is intraspecific variation in viscosity within phytoplankton species. Maybe there is a high one, but if not, whether should be some kind of niche partition between different species regarding the composition of bacteria that surround them.
Could be. I mean, if bacteria are attracted to whatever organic system we are talking about by nutrients, ingredients of nutrients, wherever you are, the MSP or some amino acids or whatever, I see little room for specificity. But there is an interesting idea that I have not mentioned, which is that bacteria could possibly be able to sense viscosity, because viscosity is changing the torque that flagella experience. So they could use the flagellar rotor as the sensor of viscosity. And if that's the case, and they have the chemotactic chemical machinery to actually detect this, that could make a distinction between which cells or what. It's an interesting.
Cool. But can you identify the bacteria? I mean, when you see the microscope,
no.
And is there any technique to do that so you can
Well, yes, it's just I'm not
just
very
put
good.
the water into and get some genetics on this?
Yes, I think so. People use live in the ocean, for example, people use microfluidic devices that will track specific bacteria depending on the chemicals they are attracted to. And we did eventually this, but we had similar devices in a field experiment that we did, in which we actually used viscosity as the attractor. But the results were not conclusive. So, and we weren't looking at the composition.
In which sense the bacteria affect the phytoplankton? You said it's good for them, but in which sense?
Well, it's very diverse, the kind of the interaction going on. The growing consensus is that all phytoplankton, especially the big ones, will have meaningful symbiotic relationships with the community of bacteria. That doesn't mean that bacteria will eat them alive if they can, but that's the consensus. And then some of these bacteria will just swim around, some of them will actually attach to the cell wall, and they exchange vitamins, things that they can identify themselves. It's a very complex.
It's very good because it will be very good to check the relationship between viscosity, the composition of the community, the bacteria community, and then the function in some way, like how it.
Yeah,
That's very good.
I don't have the you are interested.
And you said that you had a small peak of nutrients in the middle.
Bacteria, yes, that is what it's called.
Yeah, when you just had nutrients.
It's a volcano effect.
Okay.
It's basically what happens here. You have to imagine this, no?
Yeah.
So you have to imagine you have the phytoplankton cell here, and there's a gradient of nutrients around, okay? And bacteria will swim around, and then they will detect it, and they will go for it. But in many occasions, they will shoot it off. And then before they realize, oh no, now I'm going in the wrong direction, it will take some time. And so what happens then is that actually the peak in concentration is somewhat away from the cell, which is why it's the fact that increased viscosity allows bacteria to approach is a very.
I have a question about this graph that goes in the direction of what Paro asked, which is in these viscotaxis that you see in the blue line, you have no nutrients, but still the bacteria are going towards the higher viscosities. Because you have all the parameters of the motility. So my intuition is that if you in higher viscosity, the rotational diffusion is lower and the tumbling angle is also lower, then I tend to go to orient my motions in the direction of the maximum viscosity. That's a bit of my intuitive. I tend to go more straight.
Yeah.
And
there are some recent papers that this could probably.
Yeah, but my question is like if you know if this behavior, this accumulation is a passive accumulation, so it's passively induced by the medium, or if there's an active this decision.
In this model is totally passive.
Okay, this is a model.
Yes.
I thought it was an experiment. Okay.
This is an IBM model.
Okay.
And in this model, I'm not actually modeling the behavior, the interaction of the cell with the nutrient gradient. I'm just looking at, I'm just using this these functional relationships.
Mhm.
So it's kind of
Yeah, so it's coming. You input these parameters in the
yes.
in the,
I'm not looking at how, if you have a gradient of viscosity, how it will affect the distribution of forces along the cell, for example, how it will affect the function of the material.
yeah, but the accumulations are purely passive.
Yes.
I have another type of question, which is you introduced the Paxson law. So you have a graph in which I understand from the graph is that there is a theoretical law describing the viscosity, but your data overshoots this law by well, with the which
We want to
It's
know
not-
what the taxonomy is.
no, I mean, I don't know if I mean, it's
And
long to
that's a lot.
explain.
It's a law that it right? Yeah. So this is the taxonomy here. And this is the actual average viscosity that you measure. The taxonomy law accounts for hydrodynamic interactions between the beads and wherever surface you're working. So it is our right to make sure that whatever we observe is not limited.
Okay, so it's the viscosity of something which is not anything, any substance.
It's not, it's an apparent viscosity.
Okay,
It's a
yeah.
decrease in the motility of beads because of hydrodynamic
Yeah.
interactions as they get close to the surface. So if you were not aware of this, you would still be thinking this.
Usually I would think that there is okay.
Okay. Thank you. Something else?